Hey, hello guys, this is me again, Selim, and today we're gonna go again into the scGEAToolbox to do some analysis. And as always, we're gonna start typing scGEAToolbox in the command window, and we're gonna wait for start. And right now, I'm gonna continue with the process I started in the previous video, which was the data pre-processing. And the one I'm gonna take is with no QC, the cancer, colorectal cancer, and the polyp unaffected. And this was it. If you remember, we had clusters from k-means. Sorry, it's taking time. These many clusters, quite some. And we have the batch ID, and we also have the different cell types in this dataset. I am not really happy with this, so we're gonna do another method I have in my repository. You already downloaded that and installed it. I'm gonna just show it again, and it was this, the scGEAToolbox adds-on, and the two functions we are gonna use is the umap from Python, and we are going to use the latent cluster sparse. So the first thing is that we need to save it, but instead of a math file, now in the workspace. It's gonna save just the SCE checkbox on, and it should show here as SCE object. And if you hit, you can close this for now, because it's already in the RAM, in the memory RAM. So if we do SCE, it should pop up everything we have in the SCE object. We have X, the clone matrix. We have G. Let me try to make it bigger. Oh, I couldn't. So we have G, the genes, and we have S from the embedding, C for classifiers, and we have some others like cell ID, cluster ID, cell type TX. Those are the tag names we were accessing before, but this is now in the object. So what we're gonna do in this case, we're gonna do the latent clusters. The function is called like SCE equals latent. Oh, wait a minute, it's not included. If for some reason it's not included, that function, you gotta go here into the directory, right click, and remove everything, and we're gonna add it again with all the subfolders. So latent clustering annotation should pop up now. SCE equals latent clustering annotation. And if you have questions how to call it, you can double click until everything is highlighted, right click, and open latent cluster N. N is for annotation, and this is the main function. How it looks is a Python wrapper to call latent clusters, and you call SCE, the main object, and then you do resolution. I usually do 2.0, but it's good from 0.5 to 2.0, depending on how many clusters you want. In this case, I'm gonna select 2. No, it's I don't have many cells, so I'm gonna select something like 1.0, and that's it. I will not use the highly variable genes. I will use a species as human, and then method, I don't think method matters, is gonna select the KNN, the K nearest neighbors in this case. So reviewing quickly, it's gonna use for species, let's look for it, Control F, species, we have the toolbox method, that is Dr. size implementation, and okay, it accepts human and mouse. Then it's ready to call. So we do this. It's gonna fail once, and then we run it again. Oh, somehow I didn't configure it. If this same error happened to you, what you gotta do is go to this line, and oh my god, this is the outdated. I see, my bad. There is an error. I have to call a wrapper here. So let me just fix it, and I'm gonna push that change and let you guys know. Wait me a second. Okie dokie, it's fixed, and I'm gonna push it. So you guys have to upload again, upgrade the package. I recommend to do that because sometimes we have bugs and we gotta be aware of it. We can notify that as well. So I had to initialize the Python environment with this function, and that should be good to go now. So it initializes and shows the Python. Now it's gonna do a strategy I developed, which is gonna bench. It's gonna partition the number of cells depending on how many and how much memory RAM you have available, and it might take time. This time didn't took that much. Didn't even do the strategy for partitioning. Now it's doing the latent clusters, 15 different clusters identified and annotating with human. So now we can go back. After it finish, it's gonna store the results in the SCE. So we can type in scGEAToolbox, parentheses, SCE, closing parentheses, SCE. And this is how it looks. In this case, we have the different method for getting clusters. And this is how it looks. I like better the latent clusters than other methods. What we're gonna do again is reannotate. It depends on many things. If you are not satisfied, if you believe some cells are not properly identified, you can redo this step several times until you feel confident about that. All right, this case, it looks very similar. It is prior knowledge I have that some cancer cells behave like neurons, but still we need to look at markers and be very careful how we classify those. Right now, what I'm gonna take out, I think we can take out the neurons. Those are not neuron cells, but are classified as that. We gotta figure out what those are, but we can work meanwhile with that. We can look at the batch ID and see if we have the two samples here. Okay, we don't have it. All the neurons are only in the colorectal cancer. So we look for now cell type again, and right, that's what it happens. So I'm having some little issues with this, and that's what it is. I can repartition and reannotate all the time. So it seems that it's gonna be hard to compare the two samples because I need similar cell type. For example, if we take out some cells, we can do the select cells by class and by cell type, and alphabetic, it doesn't matter. We can just utilize neurons and take out the selected. Now we're gonna have only the neuron cells. That has to be named like that. But with this, we can see if the batches are there or not. Okay, cool. We have some neurons. Do we? No, it just swapped. We don't have neurons in the other sample. So we can look now at another cell type. We can probably look for, I don't know, let me look for it. cell type, and we're gonna subtract another cell type. We can try to take out fibroblast, select. And okay, for this case, we have two populations of fibroblast. Cell type. Great, those are fibroblasts, two different samples. Now, this is part of the downstream analysis. What we're gonna do here is we have the same cell type classification. Still, we need to verify that this is truly fibroblast, verifying the expressions. But right now we are gonna just proceed, and we're gonna work later how to verify the integrity of the cell classification. In this case, what we're gonna do is do some downstream analysis. The first one, it's gonna be here, differential expression analysis. I can also find that here and click on differential expression analysis here, and I'm gonna use this new procedure, use this temporary location, and it's gonna override that and get the results there. So I'm gonna compare these two samples, and the order is gonna be cancer versus A014. It's gonna take some time, so I'm gonna stop the video. After it finishes, it's gonna ask additional analysis. Sure, let's do it. It's gonna do enricher, generate volcano plot, and open output. Okay. And it's doing it only for the fibroblast. Don't get confused by this left-hand side. It's working on this other. Then it's gonna take time, gonna stop it. Okay, it finished. Then we can open the directory of where the results are stored. It's gonna be this spreadsheet, and from it we can look all the results. But meanwhile, I wanna come back here, hit OK, and it should show the volcano plot. It's over here. Let me help you to interpret this. How this can be read is like this. Whatever is gray is not significant. Whatever is blue is downregulated. Downregulated is the direction of the samples. So the downregulation happens in the A014. That means the expression of those genes is larger in that sample. And the upregulation are the yellow, and the expression is larger in the CRC sample. For example, one of the most pronounced results is here. GPX2, that's one differential expression in the fibroblast within this CRC and A014 samples. This is another gene. That other, and this other. This mitochondrial might not be good. We can just be cautious when those genes come out. But there might be some other good genes. And so forth, all these are important, but sometimes are too many. In the downregulation also can happen something, and we are not aware about what. So we got to investigate as well what's happening. Well, this is the summary of the height of those. That's to summarize the spreadsheet. And this has this much results, all the genes. So you're gonna have a gene name, the p-value, it's how confident it is. It's gonna be confident if it is within 0.05 or below. And usually we look only at the p-value adjusted. So we look for 0.05 below for the p-adjusted value. So we look at all processed, and this is what we find to be important. Then the next step is the upregulation. As you saw in the volcano plot, let me bring it again. We found MTCO3 here is one of the largest upregulated GPX2, which is also here, and so forth. If we go to the downregulated, genes like FKBP5 is going to show up as well. And that summarizes. Here the difference is that we have the fold change. It's how much the gene expression differs, and the p-value accounts if it is statistically significant, accounting for all the cells. Those are single cells. And we can look at the next note, and it mentions all this, just to be aware what every single of those quantities means. And what is in the next tabs is the differential, is the enricher analysis. For example, I can go to GOBP. That's one of my favorite databases. And you can look that this guy has a lot of genes. God, it's a lot. So that might mean something, and crucially something here. And then our job now is to look, try to link this gene, these pathways with these genes to the already published papers somebody else have done, and if it has some reasoning behind in the fibroblast disruption within cancer and the unaffected polyp tissue. I don't see, regulation of canonical WNT pathway. That's a very important pathway in colon tissue, in colon, oh my God, why is going away? In colon samples is very important mechanism to maintain the gut health. So negative regulation indicates that it's potentially bad. And this SOX9, I seen it in Axin, and SOX9 are related to cancer tissue. So what we're gonna do in this case, it's we're gonna go to the upregulated and grab the 250, and we're gonna copy it, and we're gonna jump directly to the Enricher website. Enricher. It's gonna be the first one. We hit the net. We copy paste the list, and we go to ontologies. Then GOBP is biological processes, and it's the exact same table we found before. So if we hit on table, we should be able to look at the same pathways and the same order we found here. If I look for negative regulation of WNT, it was the rank 16. If I got a 16 here, it's over here as well. It's the very same database. It's p-value adjusted, and that's great. And here are some other functions like clustergram. Clustergram, it's tricky because it cuts the pathway name, but you can ask for sum, row order sum, and then cluster, and then I want the p-value, for example. We can do c-score, maybe something different comes out, and all these RPL are coming out. That's what it is. We can do sum, and we can do cluster, and that's gonna arrange it in different order. So in this case, it's not helping much, but it is what it is. We have all these data to be visualized here, but also here, it's just in table. I'm gonna show you that it's gonna come out naturally the paper that was published originally, this data. We can put colorectal cancer, colorectal cancer, fibroblast genes, and you can just copy the genes, and you're gonna see that papers associated to those gonna come out. SOX9 blocks intestinal differentiation in colorectal cancer. So this is simple because I had the experience of that. Those genes are important in this context. So if I do Axin, very likely the main paper is gonna pop up. Colorectal cancer didn't pop up. Even Gemini suggests this gene axis plays a crucial role in colorectal cancer. Great, we know it. We expect it to be expressed in fibroblasts through function of tumor suppression. It's related to that. Great. Results about gene axin 2. Okay, this is something else. I'm just looking for papers. Overexpress in fibroblasts, blah, blah, blah. We look for axin. And, okay, it has here that they utilize it to pull down and detect interaction within PLN and axin in the caps. So that's an important gene. And that's part of the downstream analysis so far. And this is for differential expression, and I don't have anything else to add than, hey, you just look at it, and this is exactly what you want to look. It's hard because I didn't curate properly the cell type annotation here. So we gotta be very careful how we annotate. It's gonna become even better. Probably that's why I got these RPL genes. We don't know until we curate and do a couple times this process and verify that the integrity of the classification is proper. And for now, this is the differential expression downstream analysis, and I hope you catch something about it. It's complicated because you need knowledge about the sample and the cell type to get some insights of that. And that's it for me, and I wish you have a nice day. Bye-bye.